All right, page 62, 62. If you're still on your phone, it's time to get off your phone, turn to page 62.
Thanks for stopping by. This is
Okay, here we go.
2-Minute Classroom, and today we're talking about RNA, specifically mRNA, rRNA, and tRNA. Let's start with mRNA, or messenger RNA. So named because it acts as the messenger between DNA and protein production. And if you want to know the difference between RNA and DNA, I have a great video for that linked here and in the description. When you discuss RNA in school, you're usually learning about mRNA. During transcription, DNA is transcribed into mRNA. This mRNA is then translated into protein. It serves as the genetic template of a protein since the DNA can't leave the nucleus. Next we have tRNA, or transfer RNA. This is my personal favorite, and honestly one of the most fascinating molecules in genetics. RNA is generally single-stranded, but tRNA actually folds back onto itself to form a distinct T-like shape. Without going into too deep, transfer RNA is responsible for bringing amino acids together during translation to form the growing peptide chain that will become a protein.
Okay, so who brings the amino acids to the ribosome? tRNA, okay? And then they have a little 3-letter code that matches up with the 3-letter code on the mRNA. So down here we have U and A and G. So who pairs with U? A. A pairs with U, and G pairs with C. What do we call those base pairs that pair with each other very nicely? They are complementary, yes. Does anyone remember what we call the 3-letter code on the mRNA? The 3-letter code is called a codon, okay? And its opposite partner, the 3-letter code on the transfer RNA, is not a codon but an anticodon, okay? So we have the codon, we have the anticodon, and that's how they know which amino acid to bring.
And last but not least, we have rRNA, or ribosomal RNA. This RNA is the main component of ribosomes, which I've discussed in a previous video. rRNA combines with special proteins to form ribosomes, which then read mRNA to form proteins. There's a lot in that sentence, so feel free to jump back and listen again if you need to.
Okay, do you guys get that? Basically, you don't need to know much about rRNA. It just helps make up ribosomes. That's it. Okay?
But essentially, ribosomes contain rRNA and help build proteins. To bring it all together, sections of DNA are transcribed into mRNA, which is brought from the nucleus to the ribosome, composed of rRNA and protein. The mRNA is then read by the ribosome, which brings the tRNA in with the appropriate amino acids to build whatever protein the mRNA is a template for. Now watch these related videos.
Okay. So that was a very quick review, and I think there is another one here as well. This one is 5 differences between DNA and RNA. Before we start this video, see if you can tell your partner however many differences between DNA and RNA that you can. What is different about DNA from RNA? Okay, let's see what they have to say and see how many you got.
Thanks for stopping by. Let's talk about the differences between DNA and RNA. So DNA is usually double-stranded.
Why is it not going full screen for me here? Hang on. Let's try that again. There we go.
Two separate strands that are connected, which look like this, while RNA is single-stranded most of the time, looking like this. DNA is much larger than RNA, with usually millions of nucleotides. And RNA is, of course, smaller, with hundreds to thousands of nucleotides, and sometimes tens of thousands. DNA has a deoxyribose sugar, that's why it's called deoxyribonucleic acid. Its sugar structure looks like this, and in DNA, of course, it has the phosphate group and the nitrogenous base. And this carbon here is missing the oxygen, as you'll see with our RNA, which has a ribose sugar, looking like this, again with the phosphate group and the nitrogenous base.
Now, do you need to know the word deoxyribose sugar? You kind of do. If you're doing enriched, you should know the actual name, but what could you call it if you don't? D sugar. And ribose you could just call R sugar.
And this carbon here, the number 2 carbon, has an oxygen attached to it. The location within the cell for DNA is primarily located in the nucleus, with some DNA being in mitochondria as well. For RNA, while some is in the nucleus, most of your RNA is in the cytosol and in the ribosomes throughout the cytosol. For your nitrogenous bases, DNA has 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. And RNA also has 4 bases: adenine, guanine, cytosine, and uracil. So RNA does not have uracil, does not have thymine, but has uracil instead.
Okay. So, could you guys try and fill in this little paragraph here? Okay, try it on your own, and then we will talk together about what those blanks should say. See if you can fill in those blanks. Michael, see if you can fill them in. If you get stuck, ask your partner. And if you're still stuck, ask the person in front of you or behind you, or me. Is RNA a monomer or is RNA a polymer?
Polymer is like monomer is like one. So it's just like RNA is smaller, so it makes it a monomer. Also, it's like polymer.
Okay. Let's try and fill it in together. If you haven't quite finished, that's okay. We can do it together. RNA is what kind of mer? It's a polymer. It's made of many monomers stuck together in a chain. The monomer for RNA is called a nucleotide. That would be just like who? Just like DNA. Each nucleotide is made of 3 parts. What are those 3 parts? Phosphate. Next one is a sugar. And the third part is a base. If you want to call it a nitrogen base or a nitrogenous base, that's fine, but you can also just call it a base. In DNA, the sugar is D sugar. If you want to give it its full name, that is deoxyribose. But in RNA, the sugar is R sugar, which is ribose. In DNA, the bases are A, T, C, and G, but in RNA, the bases are A, U, C, G. So it is the T and the U that are different from one another. DNA is what kind of stranded? Double stranded, whereas RNA is single stranded. Excellent. And then we just get into some like really kind of nice little pictures of things. I don't know that it's particularly useful, but we can see that we have the uracil base instead of thymine. On our next page, what is RNA? RNA is a nucleic acid. Differs in 3 main ways, but you could give more than that. Single-stranded ribose sugar and uracil replaces it. You could also talk about its size and its location. Tell your partner the 3 types of RNA and what they each do. Turn to your partner. What are the 3 types of RNA and what do they each do?
Oh my yeah, before I'm sorry, I came up with this.
mRNA sends the information for a protein from the nucleus to the cytoplasm, to the ribosomes. rRNA, what does it do?
So it assembles the amino acids in the protein.
rRNA, yeah, it forms part of the ribosomes, and tRNA does what? It delivers the amino acids to the ribosomes. So you can see here, we're comparing and contrasting. So just DNA, 2 strands. Just DNA, deoxyribose sugar, just DNA has thymine. Just RNA has 1 strand, not 2 strands. Just RNA has ribose, not deoxyribose. And just RNA has uracil. But both DNA and RNA are nucleic acids. They are made of nucleotides. The nucleotides all have sugars, bases, and phosphates. So looking at that little chart there, what are 2 other major ways that DNA and RNA differ? There's 2 things that are missing from that little chart. See if you can figure them out. What are the 2 differences between DNA and RNA that are missing from that chart? DNA is longer or larger, and RNA is smaller or shorter. The next one, so we can talk about the size and we can talk about the location. Where is DNA found? Nucleus. Where is RNA found? RNA is found both in the nucleus and also the cytoplasm. Two other major ways that DNA and RNA differ. We did that. Length and location, long and short. Okay, so here is a little picture so you can actually see cytosine. One thing I'd like you to notice about this, do you see how cytosine is kind of like a short little molecule? Guanine is longer. Adenine is a longer molecule and uracil is a shorter molecule, which means that when they pair up, if I have adenine, it's a longer base, it pairs up with uracil, which is a shorter base. Then if I have cytosine, which is a shorter base, it pairs up with guanine, which is a longer base. And that means that the length, the distance between here and here, this is going to be the same distance. This is more important actually in DNA than in RNA because it means that all of the rungs of the ladders are the same size. You don't have some that are two long ones coming together or two short ones coming together, in which case you'd have a weird looking ladder, right? Where some of them were big and some of them were small. And if we look at thymine and uracil, they're almost identical. You can see we actually have this little oxygen at the top. We have an oxygen coming out the right-hand bottom side. We have a nitrogen group there. We have a nitrogen group on the bottom. The only thing that is different about uracil and thymine, and you don't really need to know this, this is just sort of for your interest, is that thymine has this extra little doodad on here. It's called a methyl group, okay? And that's it. This is just a summary chart. What's different? Strands. Ooh, we haven't talked about shape yet. That's a new one, yes? I'm going to put a star here because this is the most comprehensive of the charts. You need to know the differences and similarities. So that's something that should go on your cheat sheet. They both have the sugar phosphate backbone. They both have nitrogen bases. They have the same, they have different strandedness. They have different shapes. DNA is the double helix, whereas messenger RNA is just a single strip. tRNA forms the shape of a T, and ribosomal RNA, who knows what it's doing? It's forming the shape of a ribosome eventually, but I think that's a bunch of ribosomal DNA stuck together. Sugar, deoxyribose and ribose. Bases, thymine, uracil. Location, DNA always in the nucleus, RNA, nucleus and cytoplasm. DNA is very long, RNA is short. There's only one type of DNA, but there are 3 types of RNA. And this page, you don't need this page at all. This is just if you like geeking out on looking at chemical structures, you can be like, okay, if I talk about my nucleotide, the only thing different about the DNA from the RNA is that this is D sugar and this is R sugar. Okay, that part at the bottom is something that's useful. Okay, the nitrogenous base, A, T, C, G. The nitrogenous base for DNA is A, U, C, G. But if you want to zoom in closer, you can say, okay, well, now I'm looking and it says, oh, I got pentose sugar, deoxyribose, and I've got ribose here. That's more exciting. It shows me a little more detail. If I want to really know what's going on, I can look at my deoxyribose sugar and say, oh, look, that oxygen is missing. Yes? And if I look here on my thymine has this extra group and uracil is missing that group. Do you need to know that? No, you don't. Okay, it's just kind of there for fun. And I know some of you are thinking, wow, Ms. Spence, you have a really weird idea of fun. But I love this kind of stuff. I'm a huge biochemistry dork. What is the name of this type of RNA? mRNA. What is the name of this type of RNA? rRNA. And what's the name of this type of RNA? Well, it actually says right there it is tRNA. You'll notice that we have the words codon and anticodon. Where do we find a codon? What molecule has a codon? Yeah, so the codon is on the mRNA. And the anticodon is on the tRNA. And if you have trouble remembering which is which, well, anti has the letter T in it, so it's tRNA. Transfer RNA not only has an anticodon, but it also has something else that's very important to its function. What's the other thing that's attached to the tRNA that's really important? The amino acid. Okay, so the amino acid, the tRNA, brings the amino acid to the ribosome. Okay, and then we just have more pictures. And it's kind of cool because you can see how the transfer RNA takes on that T shape. You know how DNA, the bases match up in the middle? Well, RNA actually does that with itself. You end up with complementary base pairing. So over here you might have an A, and over here you might have a U. Over on the left you might have a C, and over on the right you would have a G. And then if we had a U on the left, on the right we would have an A. And if we had a G, then we would have a C. Okay, so it sticks to itself. Yes. Pardon me? Like that? Oh, like this away? Like that? And like up here? Yes, I can definitely do that. No worries. My battery is running, but I'm plugged in. What is wrong? Oh, because I'm not plugged in, because I'm a doofus. Okay, that's all right. I can undoofusify myself. Okay. Where's the plug? Oh, it's in here. There we go. That's going to make a big difference. Hard to be plugged in if you don't have the cord. Okay. All right, so now there's some exercises on pages 67 all the way through to 72. So you guys can work on that. You may go to the washroom, yes.
Hi. I was wondering if I could make my cheat sheet online.
You can make your cheat sheet on lined paper, absolutely.
Thank you.
And do remember, if you can't fit everything on the one sheet, I'm okay with a little bit more.
You knocked it back. Oh, I saw that. You made how much I love my back. Totally. Full fun. Right in front of me. Sure. Alright, one thing I like to do if I make cake is I like to plan out when what's going on why.
Yes.
So any laugh on. So feel like I got my day back and so I was in and then that's yeah. Do I have only rule of no so do I have so much more because I was going to do that one. You have this. Oh yeah.
Platforms, cytoplasm, pores,
Make my life visible at all? No.
organelle, ribosome, sugar nucleolus, cytoplasm. Yeah, no more outside. Because a lot of them are there. Deoxyribonucleic acid. Amino acids.
Kind of
Proteins.
well, then we went out. Oh, let everything in and out. Out and also so they will
RNA. Cytoplasm, nuclear pore, ribosome, RNA. It feels like there's a lot of room, but ribosome bugs, small subunit. Synthesis, mRNA, that's what the translation polypeptide protein. Those? Cool. This is the document it's that's all.
Because they
Use the grammar function. Yeah, for sure. Enzymes, polymers, circle proteins. Things. Yeah, absolutely. Yes. Okay. Sugar and RNA.
Kind of make it roll with this and keep it running. Isn't running on time. You do not need to know email. No. Starts with an N. Yes. A little bit. Kind of like the spell, maybe the spell. Probably from this range of question. No, like well, that was the story. Yeah, that was. I think they had oh, you. Oh, show you for yes. Oh, so we've got a survey happening here. QR code being signed. That would be great. Just help us out here. Okay. Does anyone have any questions while we're coming along here? Yeah. Hello. This is a nitrogen base. I know. It's okay. I wouldn't give you a question like that on a test. Yeah. Cool. How does the ribosome know the sequence of amino acids to go? Well, what's it reading? Oh, right. It's like the what's going through ribosomes by this is most sense. Yes. Okay, the sugar from RNA. RNA starts with R. R. Yeah. Oh, folks, make sure you have whatever paper you want with you, because there is a slight chance that we may have
snow tomorrow. It could also be rain and it won't matter. But if we have snow tomorrow, there's a slight chance that it could be a snow day. So make sure you bring whatever paper you need with you. If we don't have class, you are simply to work on your cheat sheet. Okay? You can write anything you want. Pictures. Pictures are great. Yeah. Yeah. Cheat sheet can have anything as long as it's drawn or written by you. Okay? In for as long as semester 2, you would apply to money to get through it all, which makes me really sad because I love the next grade. So if you are interested in that, most of it, right? Process here, we're not naming it in this one, way more time. This process here is called mitosis, and it's how cells start to be now, I think part of the goal like really? Yes. And I'm not in a big hurry to do anything else today because my other block missed all of last day's class. So they have to get caught up, and I don't want us to be writing 2 different tests because that's just stupid. Oh my god. Yes. When they announced it, there was such a big cheer. I missed the second part of the announcement. I was ready to leave the building, and then I went over to get it from here. I'm like, let's go. So she's like, no, we have a meeting in the library. What? What? You're gonna have to say to leave? Because they said everybody get out of the building, and I'm like, oh, everybody? It's big building. They should have moved us today, like they would have, they have to have you and goodwill that flash. Right? Morale boost that would have been. Yeah. See me, I like it's when did I let you guys know? Because I guys want 10 or that. The message that they gave us was that we would be starting at 9:30 if everything was set. Yeah, and we're like, and then at 10 they're like, okay, first we went away. They was 45 to 8 students, right? But there's some students who went right away and then transit. If you don't mind it, like when I got there. Oh, yeah. All the cities. Oh, maybe there'll be a yeah, you're right.
You make something on that, like brain call it's
no longer. Like, you know, if weather, okay. Right? If you know, the gods of plumbing, you frozen pipes. Stay off, stay warm. I think gods of it's like gods of the plumbing. Swimming in the Oh my god. Years. How is this? Yeah, yeah, yeah. Oh, and they were in the garage. It's really was. Oh my.
Wow, well that was She's not saying I find it so like coffee. I was still at home. That's crazy. I don't think so. What do you think that is just video? Me. Yeah.